Hello, and thank you for attending today's webinar, Streamline Biopharmaceutical Manufacturing with IDT Biologika's Versatile Platforms, presented by IDT Biologika and Fierce Biotech. I'm Andrea Anderson, and I'll be moderating this webinar. Before we get started, here are a few housekeeping notes. Additional resources are available in the handouts window. If you close one of the windows and need to reopen, click the name in the top navigation bar. This webinar is being recorded and will be available on demand within 24 hours. To learn more about our speaker, head on over to the speaker bio window. And finally, there will be a Q&A session at the end. If you have any questions for our speaker, you can enter them anytime in the Submit Questions window. Now, I'd like to introduce our speaker, Matthias Kahl, Head of the Development Unit, IDT Biologika. Matthias, the floor is yours. Please go ahead.
So thank you for this kind introduction and welcome everyone to our webinar. So as named, my name is Matthias Kahl and I'm the head of the development unit at IDT. And today I'd like to introduce you my company and how our versatile manufacturing platforms are helping to streamline biopharmaceutical production. From this helicopter view, you can see our state-of-the-art facility, which is able to support complex manufacturing needs from early clinical development to commercial scale production. At IDT, we bring pharmaceutical solutions to life with passion. And this isn't just a slogan, it's our mission and it's the foundation of how we work. Our teams are dedicated to delivering high quality, adaptable solutions that meets the evolving needs of our partners and patients. So let's take a closer look on how our approach can benefit your project. In this webinar, I would like to take you on a journey through the world of biopharmaceutical manufacturing and how IDT Biologika support its partner from early development to clinical to commercialization. We will start by looking at the key challenges in biopharmaceutical production, especially the complex path to clinical evaluation and CMC readiness. These steps can be challenging, but with the right strategy and support, they can be overcome. Among other, by IDT's versatile manufacturing platform, and I will explain you how we can support to overcome these obstacles and bring projects to success. Hereby, we will also explore how our integrated services and end-to-end platform solutions provide support across the entire value chain, from development through to product delivery, helping you navigating regulatory, technical, and operational complexities. To bring this to life, we will also share real-world success stories that highlights how our expertise and collaboration have helped partners achieve their goals. And finally, we will show you how we manage projects efficiently and seamlessly, ensuring transparency, speed, and quality throughout the process. So let's dive in. So at first, let's take a closer look at the key challenges in biopharmaceutical manufacturing, particularly to the journey to clinical evaluation and commercialization. One of the most challenging aspects is GMP compliance. Only across just the USA, the EMA, and Japan, there are approximately 8,000 pages of regulatory guidance. Successfully navigating regulatory complexity requires a deep understanding of clinical phase-specific requirements, the unique demands of different product classes, and the ability to manage country-specific regulation. This isn't just meeting standards, it's about anticipating them and building compliance in every step of development of new products. Equally important is also quality control excellence. Robust analytical methods and validated release assays are essential for regulatory approval and long-term commercial success. At IDT, we focus on developing technology-driven quality solutions that ensures consistency, reliability, and confidence in every batch we produce. Another important aspect is supply chain scalability. As projects move from clinical evaluation to commercial production, the ability to scale efficiently becomes essential. This requires strategic planning to meet increasing material volume demands while maintaining quality and timelines. At IDT Biologika, we are able to optimize supply chains to ensure a smooth transition, minimizing disruptions and delays in the development by our end-to-end solutions. And last but not least in my short excursus, it's technology and materials. Choosing the right raw materials and technology platforms early in development is crucial. This decision impacts not only scalability, but also regulatory compliance. By selecting GMP-compliant materials and scalable technologies from the start, we help our partners to avoid costly changes later in the process and ensure a consistent, high-quality manufacturing approach. Together, these elements form the backbone of a successful development strategy. At IDT, we are convinced to deliver the right answer to this demand and bring pharmaceuticals to life with passion. This guiding principle reflects our commitment to innovation, quality, and partnership in everything we do. We are a global biopharmaceutical contract development and manufacturing organization, specializing in a wide range of modalities, from cell culture and microbial-based proteins to vaccine, gene and immune therapeutics, biosimilars, and other sterile injectables. And since 2024, we are also proud to be part of the SK Bioscience family, which strengthens our global reach, but staying as a fully independent CDMO company. At IDT Biologika, excellence isn't just a goal, it's a standard we uphold across every aspect of our operations. This slide highlights the pillar of our commitment to delivering world-class biopharmaceutical solutions. With our over 100 years of our experience in biopharmaceutical manufacturing, we bring deep expertise and proven reliability to every project. Our end-to-end support spans from early process development through clinical trials and into full commercial production, and ensuring continuity and quality throughout the product lifecycle. We operate under the highest regulatory standards, with approvals from leading authorities, including EMA, FDA, and ANVISA. And this global compliance framework allows us to serve partners across multiple markets with confidence. In the past decade alone, we have invested more than 400 million euros into expanding and modernizing our facilities, demonstrating our long-term commitment to innovations and scalability. Our efforts have not been unnoticed. We received repeated honors, including CDMO Leadership Awards in 2025, recognizing our excellence in quality, reliability, and customer satisfaction. And beyond business, we take pride in our social responsibilities, creating long-term values not only for our partners, but also for the communities we are served for. A closer look on our end-to-end capabilities is shown on this slide, supporting your product from early development through to commercial supply. We provide comprehensive capabilities across the entire value chain, starting from process and analytical development, followed by drug substance manufacturing for both cell culture and microbial-based systems. And from here, we move into aseptic fill and finish, labeling and packaging, and finally, quality control and analytics, ensuring that every step meets the highest standard of safety and compliance. Our expertise spans a wide range of modalities. For example, in process development and drug substance, we work with cell culture and microbial-based proteins, including recombinant proteins, monoclonal antibodies, enzymes, and biosimilars. We also support vaccine development, covering viral, bacterial, live attenuated, inactivated, recombinant subunit proteins and virus-like particles. In the field of gene and immune therapies, we handle complex vectors like AAV, lentivirus, adenovirus, herpes simplex virus, and other viruses, providing tailored solutions for advanced therapies. When it comes to drug product manufacturing, we can round the value chain for our produced products in the DS, but also offering independently filling of sterile injectables for both large and small molecules. This capability and expertise is proven by our 100 years tradition across key therapeutic areas and manufacturing capabilities, demonstrating why we are a trusted partner for both clinical and commercial scale projects. For example, for vaccines, we have supported a wide range of reference projects, including Zika, chikungunya, dengue, RSV, HSV, Ebola, and COVID-19. We have also been a reliable partner for governmental organizations delivering emergency use vaccines for more than 2 decades. In the area of gene and immune therapeutics, we have worked on pioneering projects involving oncolytic viruses such as herpes simplex and measles, as well as viral vector-based gene therapies like AAV. We are also proud to be the exclusive CDMO partner for one of the first commercial oncolytic virus products, underscoring our leadership in advanced therapies. Our capabilities in aseptic filling of sterile injectables are equally robust. For over 35 years, we've been a trusted supplier to customers worldwide, handling both large and small molecules. Our expertise spans vials, also here for both liquid and lyophilized, as well as pre-filled syringes. Reference projects include IDT biosimilar, commercial medical Botox, Ebola, and RSV vaccines, and gene therapies in clinical development. And to give you a sense of our scale, we have manufactured over 4,500 batches, GMP batches, in just the past 4 years, demonstrating our reliability, consistency, and capacity to deliver. Now let's take a closer look at one of our key manufacturing lines, the Drug Substance Building 200, which is our solution for bacterial production for both clinical and commercial supply. In-house, it houses over 3,000 square meters of clean room space, classified as Class C, B, and A, ensuring compliance with the most stringent GMP standards. Within the building, we operate 8 specialized production units, as you can see on the floor plan, covering bacterial and vaccine production, as well as buffer and media preparation. This modular setup allows us to tailor operations to the specific needs of each project. Our technology portfolio includes 800-liter stainless steel tank bioreactors, isolator, and 6 200-liter single-use bioreactors, giving us the versatility to handle a wide range of processes and scales. We have also adaptable downstream equipment, for example, for clarification, tangential flow filtration, and chromatography, which can be configured based on the unique requirements of each product and process, but it's not listed here in detail on the slide. The second manufacturing building I want to introduce is the Drug Substance Building 205, which is dedicated to mid-scale mammalian production for clinical and commercial applications. The facility features a 900 square meter cleanroom area classified as Class C, B, A and B, ensuring full GMP compliance for sensitive biologics and advanced therapies. The building is organized into 4 independent suites, each designed to support a range of modalities, including viral vector and protein production, as well as vaccines, oncolytic virus, and other therapeutic biologics. The modular setup allows us to run multiple project modalities while maintaining strict segregation and process integrity through changeover controls. In terms of technology, we offer flexible platforms that include cell stack or cell factory, the iCELLis 500 fixed bed bioreactor, roller bottles, and 200-liter single-use stir tank bioreactors. And this variety enables us to tailor the production approach to the specific needs of each product and scale. As with our other facilities, we are also providing versatile downstream processing equipment, which can be configured to meet the unique requirements of each process. And this facility is a key part of our adaptable manufacturing lines, supporting the growing demand of mammalian-based biologics and gene therapies. Our newest drug substance building is the Building 203, built in 2023, covering our large-scale mammalian cell line production capacities, designed to meet the highest demand of commercial manufacturing. This facility features a 1,700 square meter cleanroom area, classified as Class C, B, and A, and is equipped with 5 large production units, here displayed in green on the floor plan, and 3 C-train rooms displayed here in blue on the floor plan. And this setup allows us to support high volume production while maintain flexibility across multiple projects. The drug substance building 203 is purpose-built for viral vectors, protein production, as well as vaccines, oncolytic therapies, and other modalities. It's a cornerstone of our capacity to deliver complex biologics at commercial scale or higher demands for late-stage clinical development. And similar as for building 205, we have different technologies installed here, also cell stacks, iCELLis bioreactor 500, and single-use stirred tank bioreactors, but here in a higher range volume from 50 to 2,000 liters. And this operational range gives us the ability to scale processes efficiently from late-stage clinical development direct into commercial manufacturing without any further change in production line, building, or equipment, and ensures a seamless transfer into market supply. According to the versatile drug substance manufacturing, we have also multiple fill and finish capabilities established, which completes our end-to-end service. Here operate 9 filling lines equipped with both a restricted access barrier system or isolator technologies, ensuring the highest level of sterility and operator safety. Our platform supports a wide range of formats, including 2 to 24R vials for glass vials or CZ vials, the copolymer one, and also pre-filled syringes ranging from 0.3 milliliter to 3 milliliter. And this flexibility allows us to accommodate everything from early clinical phase manufacturing to large-scale commercial production. In terms of batch sizes, we can handle volumes from 500 to 500,000 doses per batch. And here also liquid and lyophilized options are available. As an example, you can see on the right side our smallest filling line 4, a RUPS-equipped line, which is ideal supporting batch sizes from 500 to 10,000 vials. And on the other end of the spectrum, our high-speed filling line 2, operating under isolator conditions, can fill up to 500,000 vials per batch, making it perfect for high-volume commercial supply. And this universal approach ensures that we can scale with your product, whether you're just starting clinical trials or preparing for global launch. To underline this, you can see on this slide how IDT Biologika platform solutions are designed to meet the diverse scale and technology needs of biopharmaceutical productions, from early development to commercial supply. On the left, you see a comparison of various bioreactor platforms based on their typical dose range per batch. Of course, the actual dose per batch strongly depends on production yield and the specific dose formulation, but this should still provide a rough indication of potential manufacturing yields. These are grouped into 2 categories: suspension-based system and adherent systems. For suspension cultures, we offer stirred tank bioreactors, wave bioreactors, and shaker flasks, each suited to different scales and production types. On the adherent side, we utilize microcarrier-based stirred tanks, fixed bed bioreactors, roller bottles, and cell stacks or cell factories. This range allows us to tailor the production approach to the specific needs of your molecule and modality. The x-axis shows the scalability of each platform, ranging from small-scale production of hundreds of doses to large-scale commercial batches exceeding 1 billion doses. It is clear that some technologies have inherent limitations, while stirred tank bioreactors remain unlimited in scalability. However, demand and process constraints must be taken into account, and sometimes require specific manufacturing solutions, which we are fully equipped to deliver. Again, The actual doses per batch depending highly on production yield and dose formulation, but this should still provide a rough indication of potential manufacturing yields. The selection of the right technology for product development and future commercial demands is one of the key aspects for success of your product and should be considered from the very beginning of development. This is one reason why we rely on technology consistency at IDT, from process development to clinical scale and commercial manufacturing, to ensure seamless and efficient scale-up strategies. In short, our established manufacturing platforms are built to adapt, providing the right technology and scale to meet the evolving demands. But let's talk now about a real story from our customer and start with an analytical topic where our versatile analytical capabilities were used to solve advantageous virus constraints. So this case study highlights how the introduction of next-generation sequencing is transforming the landscape of advantageous virus testing and what new challenges it brings to process risk assessment. So NGS is a rapidly gaining relevance as a powerful detection method in advantageous virus testing, and regulatory guidance is evolving to support the shift, allowing NGS to replace traditional in vivo testing in many cases. Its strength lies in its unspecific detection system, which enables broad and sensitive screening for a wide range of viral contaminants without the need of prior assumptions about what might be present. However, with this power comes complexity, and the interpretation of NGS data and the design of appropriated follow-up testing are critical to ensure product safety. And this is where scientific expertise becomes essential. At IDT, we have played a pioneering role in advancing the applications of NGS technologies for advantageous viruses testing, and our expertise has helped to shape best practice and contributing to the development of regulatory frameworks. For example, the FDA ICH Q5A revision now includes NGS as a viable approach for both broad and specific virus detection. It allows the replacement of in vivo studies without requiring head-to-head comparison, and even suggests that NGS may replace cell-based infectivity assays. However, any positive result must still be too roughly investigated to determine if it reflects an infectious virus. Similarly, the European Pharmacopoeia is drafting a new general chapter, the 2.6.41, focused on high throughput screening for advantageous virus agents testing. And this chapter will provide guidance on implementation and validation strategies, helping to standardize the use of NGS across the industry. So this case underscores the importance of combining cutting-edge technology with regulatory insight and scientific rigor, something at IDT we are proud to offer to our partners. So this case study highlights the importance of scientific interpretation of next-generation sequencing for advantageous virus testing, particularly when it comes to distinguishing between true viral contamination and background signals from host cells. So in this example, NGS was used for release testing for a vaccine product, and the analyzers initially flagged a potential detection of the avian leukosis virus, which is a retrovirus. And the data showed a hit against the ALV genome, but upon deeper scientific review, it became clear that this was a known artifact caused by the use of chicken-derived cells in the production process. The reads covered only a small region, about 325 base pairs, with 2 enrichment peaks, which is consistent with the backbone signals from ingenious retroviral elements that are presented in other eukaryotic cells like the gallus. And this demonstrates the clear key limitation of NGS because while it's highly sensitive, it cannot always discriminate between endogenous retroviral sequences and active viral contaminations. That's why in such a case, a PERT assay, which specifically detects reverse transcriptase activity, is recommended to confirm or rule out the presence of infectious retrovirus. In this case, a negative result was very likely, but the scientific rigor applied in interpreting the data was essential to reaching that conclusion. So here our analytical toolbox at IDT plays a critical role in developing a robust follow-up strategy after a positive NGS result. In this case, the NGS analyzers flagged the potential retroviral signal, raised the question if there is a retroviral activity. And to answering this, we performed a specific FPERT assay, which confirmed the presence of a reverse transcriptase activity, which raised then the follow-up question, what kind of subunit or subgroup is presented? And here we used the digital PCR, with specifically detected the E subunit, which is a known endogenous retrovirus that is not considered critical for product release. But to finally confirm this, the next step was also to investigate if this subunit is infectious. And for this, we cultivated the sample using chicken-derived DF1 cells over 5 passages, followed also by FPERT testing of the supernatant, with the clear result that this subunit is not infective. And with this comprehensive follow-up strategy, we were able to confirm that the retrovirus detected was non-infectious and endogenous. And as a result, the product was safely released with no risk to the patients. And this case clearly demonstrates how IDT's scientific expertise and advanced analytical capabilities can de-risk complex findings, validate product safety, and support confidence in decision-making, even in face of ambitious or unexpected results. The next case study focused on a customer project preparing to transition from early clinical manufacturing to phase 3 and commercial production, which is, from my perspective, also a critical step that requires significant process optimization. And initially, the customer had completed phase 1 and 2 clinical trial material production at a moderate scale. And the former process workflow is here illustrated on the right-hand side of the slide. And however, as they looked ahead to phase 3 and commercial readiness, it becomes clear that the existing process would not scale effectively. Several key challenges emerged during the process development. For example, the upstream process relied here on CellStack 40 technology, which limited scalability, and particularly due to the centrifugation capacity constraints. And also the systems in use were not state-of-the-art, which impacted performance and efficiency. Another point is that there were too many manual steps, reducing process control and robustness. And as the total result, the process validation and process performance qualification became increasingly difficult. And finally, achieving GMP compliance at commercial scale posed a significant challenge due to the unrobust process design. And to overcome these hurdles, we worked closely with the customer to redesign the process and transfer it to a scalable and GMP compliant technologies, laying at the end the foundation for CTM-free manufacturing and future commercial production. So I selected this case to highlight the importance of early planning and the right technologies or technical strategy when preparing for late-stage development and market readiness. So on the next slide, you can see what we have done for the upstream process development to scale up the virus production process by introducing the iCELLis 500 bioreactor technology and optimizing also the seed train strategy. To address the higher cell demand for the iCELLis 500, we redesigned the seed train by integrating HyperStack 12 and 46 layer systems, which allowed for a more efficient and compact expansion of adherent cells. But the real transformation came with the implementation of the iCELLis 500 fixed bed bioreactor, since this system enabled us to tightly control key process parameters such as pH and dissolved oxygen and temperature, ensuring consistent and reproducible conditions throughout the production run. And as a result, we achieved here a 12-fold increase in production scale, while also improving process control and reducing manual handling in the process. And the final harvest volume here was 140 liters instead of formerly 12 liters. As this is going into the downstream processing, we also addressed here some scalability limitations by optimizing the process design and introducing more robust technologies. The original downstream processing relied heavily on centrifugation, which posed challenges for scalability, consistency, and GMP compliance in the future. And as I mentioned, to overcome this, we redesigned the process to incorporate filtrations and chromatography-based solutions. And as shown in the flowchart, the process begins with 140 liter clarified feed, followed then by DNA removal and then tangential flow filtration for ultrafiltration and diafiltration. And here, after this point, we then introduce an exchange chromatography step to enhance product purity and removed process-related impurities. After this is followed by a second TFF step, and finally stereo filtration formulation, and finally then filling, labeling, and storage. The process yield here was, in this example, 1.5 liters for the final product after sampling, and was ready for clinical and commercial use. We also evaluated hold times at critical stages to ensure process robustness and mitigate risk during the manufacturing. And this optimized downstream processing design not only improved the scalability and the integration of single-use process solution, but also enhanced process control, the quality, and regulatory readiness as a key factor for successful PPQ readiness and commercial launch. So the result of this adaptation was a successful scale-up strategy and process validation of the optimized process, highlighting at the end by consistency and comparability across production scales and technologies. To explain, let's start with the first graph on the right, which shows the titer profile across 3 large-scale iCELLis 500 batches. And as you can see, the virus harvest titer remains highly consistent across all runs, confirming the robustness of the upstream process. And this was critical for demonstrating here the process robustness, but also the PPQ readiness. The second graph on the right compares the virus titer at downstream stages from harvest to formulation. And the result show that the large-scale iCELLis 500 process delivers comparable yields to the previous CellStack 40-based production process. And in addition to the performance, the new process design introduced a simplified, closed, and scalable upstream processing platform with also significantly reduced contamination risk and aligned with modern GMP expectations. So this case is a strong example on how thoughtful process redesign combined with the right technologies and expertise can enable a smooth transition from clinical to commercial manufacturing. I hope the shown examples from our customer gave you a good insight in manufacturing complexity, but only have touched some specific points in the development. And to give you a broader understanding what development of manufacturing process means, I generated the slides that also only covering the major interactions between departments of our company. So in general, this picture illustrates the complex and highly interconnected nature of biopharmaceutical manufacturing. The target is to develop sufficient process design, which serves as a foundation for successful product development and commercialization. The process design itself is described by essential tools such as tech transfer database, which covers any process-related parameters and informations. Then we have the risk assessment via a failure mode effect and analysis, and the analytical project master plan defining the quality control requirements to your product, all of which helps ensure that processes are robust, scalable, and at least compliant. Surrounding this core are multiple disciplines that must work in harmony. So we have the analytical development, the quality control, process development, validation, technical operation, supply chain, procurement, and even human resources. So each of these functions plays a vital role, and their interdependencies means that a challenge in one area can quickly impact the other. And this complexity is one of the biggest hurdles in biopharmaceutical manufacturing, but it's also where IDT excels, since we can offer here a fully integrated in-house services that are seamlessly aligned across all functions. And this integration not only improves efficiency and communication, but also ensures that your project stays on track from start to finish. And communication between these areas, as well as with the customer, is another key factor to success. At IDT Biologika, we believe that successful project executions depends not only on technology, but also on expert guidance and seamless coordination. So at the center of our project management approach is the client program manager, and he serves as a single point of contact, providing, let's say, the project oversight, making the key decisions and guiding you through every phase of your project. They coordinate across all critical functions, for example, tech ops, supply chain, development, and MSAT, to ensure an alignment and progress. Supporting the client program management is our manufacturing science and technology team, and these are our scientific experts who bridge the gap between R&D and manufacturing. They lead technology transfer, define critical process parameter, and ensure that new products are introduced in manufacturing in a robust, scalable, and reproducible manner. And finally, we have assigned a dedicated core team in manufacturing to every project. And this team includes specialists from tech ops, quality, and engineering, who are supported by supply chain, lean management, and controlling to ensure smooth executions and reliable supply, all while maintaining close collaboration with you at the end, the customer. And together, these roles form a tightly integrated structure that enables us to deliver complex biopharmaceutical projects with precision, transparency, and speed. So to wrap up, let's talk about why IDT Biologika is the right partner to bring biopharmaceutical products to life faster, safer, and with confidence. So first, we offer innovations and technology through ready-to-use platform and proprietary solutions that accelerate development and reduce risk. Second, we have fast and seamless tech transfer that is driven by dedicated experts who ensure speed, reliability, and minimal disruptions. So at least to stay on track with your project. Third, we bring regulatory excellence to the table with a strong record of compliance with global authorities. We help you navigate complex regulatory landscapes and secure market access. Fourth, our flexible capacities and services mean you get scalable manufacturing, in-house analytic, and secured supply chain all under one roof. And finally, we believe in trusted partnership. Our collaborative approach is backed by decades of experience and a commitment to transparent communication. So you always know where your project stands. And at IDT, we don't just manufacture products, we build partnerships that deliver results. And with that, I hope I was able to give you a brief insight into the complexity of manufacturing and how IDT can support your projects and help to bring it to success. I thank you very much for your attention, and I'm now looking forward to answering to your questions.
Yes, thank you so much for that presentation. It is time for the Q&A portion of today's webinar. If you haven't submitted your questions yet, please do so now. All right, the first question, what are the key advantages of using iCELLis systems for viral vector production compared to other technologies?
Yeah, thank you so much for this question. So I think there are several advantages for the iCELLis 500 system. So I think the first thing is clear, it's the scalability, especially when we compare this with static system like CellStack 40 or Cell Factory 40. I mean, this is a key benefit. But the other one that is really important is the process control, which plays a crucial role in robust manufacturing. And with this integrated monitoring of pH and dissolved oxygen and temperature, for example, it ensures that we have a robust manufacturing and also reduce manual handling steps during the critical production phase, which was in this example, the virus propagation.
All right, another question I see here. What kind of support does IDT Biologika offer during the tech transfer process from development to commercial scale?
Okay, thank you also for this question. So yeah, I deeply believe that the tech transfer mindset here is centered around guiding our customers to from the really beginning, initial onboarding, all the way to project success. And so in our mindset, this goes beyond identifying gaps in the current let's say program that we are provided. It's more about, let's say, to define a combined strategy, a kind strategic pathway to bring at least the product to life. And I really can say that every department in our company is really aligned to that goal, and at least to ensure a fast and seamless process transfer tailored to specific needs of your product and project.
All right, another question. Can you provide examples of how your platforms have helped reduce time to market for clients?
Yes, thank you also for the question. So I can say that established platform and versatile platforms that are already existing have also specific advantage on timeline. We can really dramatically accelerate project timelines, for example, to bring it in a perspective, developing a GMP-compliant process from scratch typically takes 2 to 3 years, based on my experience. But this can be leveraged by proven, let's say, platform concepts down to less than 1 year. So, and the timeline is one critical, let's say, benefit here. But beyond speed, this platform also support regulatory compliance, since then we can already use established materials, manufacturing designs, equipment, and the facilities that are already validated and approved, and avoiding at least delays and future hiccups when it comes to clinical evaluation and commercial, let's say, launch of different products.
All right, another question. What are the most common challenges that your clients face during clinical manufacturing, and how do you address those?
Also good question. So can tell that we often work with clients, as shown in the example, who are already in the clinical evaluation, but lack a clinical or clear strategy for late stage development or commercialization. And in many cases, there are gaps in scale-up planning or having a limited understanding of regulatory requirements, especially as projects move towards commercial readiness. But for us, it's never a showstopper. And as I mentioned earlier, our mission is to bring this pharmaceutical solutions to life. So we guide our customer to success through our fully aligned and integrated services. And this really allows us to streamline even the most complex projects, since the interconnections between our department is really good and communication is short. So we making the suggestions that our customer is helping to realize their project and to overcome any hurdles here.
All right, another question. Are there any upcoming innovations or expansions planned for your manufacturing platforms?
Yes, so at IDT, as I have already also presented, so we are continuously investing in expanding, let's say, capabilities as well as the capacities, and our vision is to grow alongside with our customer projects. And over the past decade, as I have shown, we have invested more than 400 million euros. And I think this is a clear commitment to innovation and our infrastructure. And this will continue also in the future. And besides the physical capacity, we also advancing our technology edge. So as I have also shown, for example, we have introduced NGS, which was not a clear requirement from the regulatory, but with the idea to stay ahead of evolving regulatory expectations and enhance product safety at the end. And in addition, we are also driving digital transformation across our workflows, which is also including the integration of artificial intelligence in several business areas. And this is not only for improving operational efficiency, but also enables deep learning applications in manufacturing and helping us continuously improve product quality and process reliability.
All right, well, that concludes our webinar today. If we were not able to get to your question, we will do our best to follow up after today's webinar. As a reminder, a recording of this session will be available on demand within 24 hours. Thank you for joining us today, and we look forward to seeing you next time.
Thank you so much.